What specific problem does the new method or algorithm aim to solve in the context of LLMs and drug discovery?
The new method aims to address the problem of hallucinations generated by large language models, LLMs, in the context of drug discovery, specifically by exploring how these hallucinations can enhance model performance rather than merely mitigating them. The study hypothesizes that hallucinations, which often contain unrelated yet insightful information about molecules, can inadvertently assist LLMs in making predictions about molecular properties and potential applications. By systematically investigating the effects of hallucinations through modified prompts and comparing the performance of various instruction-tuned LLMs, the research demonstrates that incorporating hallucinated text can lead to significant improvements in tasks such as predicting antiviral properties or toxicity of molecules. The findings indicate that hallucinations can provide valuable insights that enhance the ILM's ability to differentiate between molecules, thereby fostering creativity and innovation in drug discovery.
Can you explain in detail how the proposed method for leveraging hallucinations in LLMs works? Illustrate the process step by step, including key components and decisions taken at each stage.
The proposed method for leveraging hallucinations in LLMs involves a systematic approach to enhance drug discovery tasks. First, the process begins with generating textual descriptions of molecules based on their SMILE strings using various LLMs. Next, these generated descriptions, which may contain hallucinations, are incorporated into the model's input, alongside the SMILE strings and specific task instructions. The prompt template is structured as SMILES, description, instruct, where the description component can be replaced with either hallucinated text or reference descriptions from a domain-specific model like MOLT5. In the experimental design, the researchers evaluate 7 instruction-tuned LLMs, comparing their performance with and without hallucinations. The key decision here is to assess how the inclusion of hallucinated text impacts the model's ability to predict specific properties of molecules, such as antiviral properties or toxicity. The results indicate that hallucinations can improve model performance, with LLAMA3, 1-8b, showing significant gains when using hallucinations generated by GPT-3, 5. Finally, the study investigates additional factors influencing the effectiveness of hallucinations, such as model size and generation temperature, concluding that larger models benefit more from hallucinations, while the temperature has a minimal impact. This comprehensive approach illustrates how hallucinations can be strategically utilized to enhance LLM performance in drug discovery tasks.
Could you provide a simple example that demonstrates the inner workings of your proposed algorithm, specifically detailing the generation of hallucinated descriptions and their integration with the drug classification task?
In our study, we utilize a molecule represented by its SMILE string, such as CC1 bridge C equals O and C equals ON, C2 CCOC COO2 C1 N equals N plus equals N, to generate hallucinated descriptions using LLMs like GPT-3. 5. For instance, the generated description might inaccurately state that the molecule is made up of hydrogen, and include subjective claims about its potential applications in drug discovery. We then integrate this hallucinated text into our drug classification task by formatting the input as follows: SMILES, hallucinated description, instruct. This setup allows the LLM to process both the molecular structure and the hallucinated information, which, despite being incorrect, can enhance the model's confidence and improve its predictions regarding whether the molecule possesses specific properties, such as antiviral activity. Our experiments show that this integration of hallucinated descriptions leads to significant performance gains in drug discovery tasks, as evidenced by the improved ROC-AUC scores.
What are the practical advantages of using hallucination in drug discovery as proposed in your study, and how do these advantages compare against traditional methods that aim to mitigate hallucinations?
The practical advantages of using hallucination in drug discovery, as proposed in our study, include the enhancement of LLM performance in predicting molecular properties and functions. Our findings indicate that hallucinations can provide valuable insights, such as high-level descriptions and potential applications of compounds, which may inadvertently assist LLMs in making predictions. For instance, LLAMA 3.1 to 8b achieved an 35% gain in ROC-AUC compared to the SMILES baseline when utilizing hallucinations, demonstrating a significant improvement over traditional methods that focus solely on factual accuracy. In contrast to traditional approaches that aim to mitigate hallucinations, our study suggests that embracing hallucinations can foster creativity and lead to innovative solutions in drug discovery, as they allow for the exploration of novel patterns and relationships within chemical data. Overall, leveraging hallucinations presents a unique opportunity to enhance the drug discovery process, making it more efficient and potentially more effective than conventional methods that prioritize factual consistency.
Describe the experimental design used to validate your method. What datasets were utilized, and what metrics were employed to gauge the effectiveness of the proposed approach?
The experimental design involved evaluating the performance of 7 instruction-tuned LLMs by comparing their outputs when provided with different types of input descriptions, specifically focusing on the impact of hallucinations. The datasets utilized were 5 from the MoleculeNet benchmark, which included HIV, BBBP, ClinTox, CIDR, and Tox21, each related to classifying molecules based on their biophysical and physiological properties. The classification task was framed as a next token prediction problem, where the input consisted of a SMILE string, a description, and task instructions. To gauge the effectiveness of the proposed approach, the primary metric employed was ROC-AUC, which measured the model's ability to predict whether a molecule possessed specific properties. The results indicated that adding hallucinations to the prompts generally improved model performance compared to using only the SMILE strings or reference descriptions.
What specific results did your experiments yield, particularly regarding the performance improvements achieved by the LLMs when utilizing hallucinations? Can you elaborate on any significant trends you observed in your findings?
Our experiments demonstrated that hallucinations significantly enhance the performance of LLMs in drug discovery tasks. Specifically, LLAMA 3.1 to 8b achieved an 35% improvement in ROC-AUC compared to the SMILES baseline and A13, 79% improvement over the MOLT5 baseline when utilizing hallucinations generated by GPT-3. 5. Additionally, hallucinations from GPT-4o provided the most substantial average performance gains across models, with increases of 4.07% and 4.54% compared to the SMILES and MOLT5 baselines respectively. A notable trend observed was that larger models exhibited greater potential for improvement with hallucinations, while the generation temperature had minimal impact on performance. Overall, our findings indicate that hallucinations can be leveraged as a beneficial tool in enhancing LLM capabilities for drug discovery, with specific models like GPT-4o and LLAMA 3.1 to 8b showing particularly pronounced benefits.